All right, I think that's about as much time as we can give as folks are still kind of getting in. I can see the numbers increasing, but we got a lot to get through today, and we want to respect people's time. So I think we just got to get started. So welcome to everybody. This is now the fourth of our series in webinars that we've been co-hosting together between Regis Technologies and GZP. The topic today is process development strategies to deliver robust manufacturing processes. We have a couple speakers we're real excited to work with you on, but before we can get started on that, let's get through the formalities. We're going to do a quick introduction of GZP, a quick introduction of Regis, talk about the speakers, and then we'll get right into it. So, Lauren, if you want to start it off.
Yeah. Thank you, Dan. Good morning, everybody. As Dan mentioned, I head up the business development team here at Ground Zero Pharmaceuticals. On here, we also have our regulatory expert, President and CEO, Evan Siegel. If you guys have any questions at the end of the presentation, feel free to ask. We will be there to answer. Just to give you a really quick overview, we are a one-stop shop strategic regulatory and product development. We offer the regulatory service, all the ECTD submissions, reg ops, preclinical, non-clinical, clinical services, medical writing, CMC, and then the project management. We work with mainly small and medium-sized companies between biotech, pharma, combination products, and med device. We work across a lot of different indications and therapeutic areas. Our sweet spot is pre-IND through IND into marketing submission. If anyone wants to learn a little bit more about our structure and the way that we do things, feel free to reach out to me personally. My information is on here. But enjoy the presentation, and I hope you guys take a lot from it.
Thanks, Lauren. And I'm Dan Weissmuller. I'm the Director of Business Development at Regis Technologies. And just to say a few words about Regis, we are a privately held US-based CDMO specializing in the development and manufacturing of novel drug substances. We're centrally located in the Chicagoland area, and we've probably served the biotech and pharmaceutical industries for over 60 years. Just my time at Regis, we've supported over 100 clinical candidates, commercialized 10 various projects, and we're currently producing 5 globally marketed drug APIs that are used in globally marketed drug products. Regis continues its mission to help expedite drugs to market by offering our talent and hard-earned experience in our project teams. The advantage of working with Regis, are those people, and working with an established domestic supplier that gives you the routinely FDA inspected brand. You get the reduced global supply chain risk, ease of communication, ease of access. But also through our single site strategy, where Regis laboratories, manufacturing facilities are centrally located on the same campus, allowing real-time in-person collaboration between our process chemistry, analytical, solid state, manufacturing, along with our clients. If anyone is interested to learn more about Regis, I'll be sharing my contact information as well, or feel free to reach out in the chat, and we'll catch up with you later. But enough of that. Allow me to introduce your speakers today. So today we're going to be speaking with Chris Castle, Eric Buck, and Ron Mueller. So Dr. Castle received his PhD in organic chemistry from the University of Iowa. After graduating, he began to work in process chemistry division of Dow AgroSciences, later Corteva AgroSciences, or AgroSciences. And after several years working at Dow, Chris transitioned to pharmaceuticals, and since 2019 has played a leading role in our process chemistry group here at Regis Technologies. Dr. Buck received his BS in chemistry at the University of Minnesota and PhD from the University of Pittsburgh. After his postdoctoral stint, working on natural polymers, he joined Fonterone, which later became Epicent, and then made his way to Regis, where he, as a senior process chemist, he quickly rose to the rank of principal scientist, then to group leader, where now he oversees the development of multiple projects as an active member of the process safety team. And then there's Dr. Mueller, and Dr. Mueller has more than 30 years of the pharmaceutical experience in the pharmaceutical industry, with over 25 years of pre-formulation and solid-state chemistry experience. After earning his BS in chemistry, he studied chemistry at Karlsruhe Institute of Technology in Germany as a Fulbright student scholar. And I'm going to it's a Quadrille Mobay Fellow. It's something I probably need to learn a little bit about. And Ron received his PhD from the University of Nebraska-Lincoln in organic chemistry. Ron came to Regis and started our solid state chemistry department in March 2019, and has added capabilities that supplement our process chemistry, and including salt, co-crystal, polymorph screening, and a number of other activities that really help bring things forward for us. And we're going to talk about that today. So I think that serves as our introductions. And if maybe Eric, I can now kick this back over to you.
All right, thanks Dan for that introduction. Welcome to our talk for process development strategies to deliver robust manufacturing processes. So what is process chemistry? Process chemistry is a development of a safe and consistent and robust chemical process. Goal number one is to develop a safe process. Chemistry is inherently dangerous, but we can design a process that takes that into account and develops engineering controls to really minimize that risk. We want a consistent process. We want to know that every time that we run something, that we know what we're going to get out at the end in the sufficient quality that we're looking for. And the goal number three, that robust chemical process, we want to know that if we run it under a certain set of conditions, that'll work every time. Here at Regis, we are focused on the production of active pharmaceutical ingredients, or APIs. And we typically begin relationships with clients for their first scale up of their API to begin tox studies and to continue that relationship all the way through FDA approval and commercialization. Just a quick slide here on the drug development lifecycle, where Regis fits into that, and the roles of a process chemist during each of the phases. Initially, for that preclinical IND application in phase one, the process chemist here is looking at route scouting, route selection, enabling routes to deliver that API material selection. As I'd mentioned, we want to develop a safe process, and this is where we start to look in removing any toxic, dangerous reagents or things like that. We are also starting to do our first batches for toxicology and into our first GMP batches. In this early phase, solid state is also looking at different form screening. As we move into phase one and phase two, we're really looking into process improvement and optimizations. Before this, we have run quite a few batches at this point in production. As we get into these later stages, we're really looking at optimizing the process. We've been through several runs. Maybe there are a few inefficient spots, or maybe a low yield that we would like to really improve. And here we're also looking at reducing step count. This doesn't necessarily mean steps to actually make the API, but it could also mean just steps in the general process, as we'll get into later. We're also looking at reducing cost of goods at this time if we can. Solid state is also looking at polymorph screening. And our clients are starting to select their final form for the drug substance or API. As we move into the later phases, the phase two pivotal studies and phase three, here's where we switch over from optimizing the process and really start to focus in understanding our chemical process. And for that, we go into ranging studies, and this is to determine safe operating ranges or proven acceptable ranges, PAR. We also look at fate and purge studies for impurities. At this point, we want to know, when are we making impurities? When are they getting removed from our process? And what levels can we tolerate to make sure that we have a sufficient quality at the end of the process? We also look into critical process parameters and key process parameters at this time. And all that leads up to our engineering batches, our registration batches, into the pre-validation and validation work. Eventually, this transitions into the NDA application, FDA review, approval, and then eventually launch. And with that, I would like to turn it over to my colleague, Ramil, and he'll give you a nice introduction into solid state chemistry.
Thank you, Eric. Pharmaceutical solid-state chemistry can sometimes be referred to as materials chemistry or solid form sciences. Solid-state chemistry can be thought of as a study of functional relationships between synthesis or process, structure, and properties of solid phase materials. Solid-state chemistry identifies materials with combination of properties tuned for specific applications. In the pharmaceutical industry, it means understanding the drug's substance properties to select the appropriate solid-state form for further drug development. Most active pharmaceutical ingredients exist in different crystalline arrangements, thus exhibiting polymorphism. Polymorphs have different physical-chemical properties, which may affect stability and solubility, consequently, the dissolution and bioavailability of the drug product. Not knowing if an API exhibits polymorphism and to what extent could range from being a nuisance, having to restart a laboratory study, to catastrophic consequences if the API is already commercialized as a drug product. Two examples are Norvir and Nupro. These type of consequences can endanger patient lives and must be avoided. Ritonavir, if you recall, was approved in 1996 with only one crystalline solid form known to exist, even with attempts to identify other polymorphs. About 2 years on the market, a new, more stable solid form was discovered, eventually causing the drug product to be removed until a costly solution was identified and then remarketed many months later. Submitting the appropriate information for an IND application, especially the solid-state aspects of the drug substance, can accelerate new drugs into clinical trials. Having the appropriate information from a physical chemical perspective will reduce uncertainties about the solid-state form, expedite the process, and openness with the FDA. Typically, the most thermodynamically stable polymorph should be identified as early as possible to reduce time to market and cost, because it has the lowest propensity to transform during the process development, formulation development, and storage. The solid-state chemistry team provides the following services: materials characterization, screening and selection studies for polymorphs, salts, and co-crystals, crystallization development, technical assessment and consultations, method development, and qualifications. The solid-state chemistry team can be used as a standalone service or as part of a comprehensive drug development program. Remember, the goal of manufacturing process development for the drug substance is to establish a commercial manufacturing process capable of consistently producing drug substance of the intended quality, as stated in ICH-Q 11. Today, you will hear how process chemistry and solid-state chemistry work as an integrated team to deliver the right solid-state drug substance form for our clients. And with that, I will turn it back over to Ed.
Thanks, Ron. So when we begin process development, it really begins with a client tech transfer. At this time, we get our client's procedure for the first time, and they can range anywhere from we have made a few milligrams to we have made several kilos. Typically at this time, we can start to see that several of these processes have just some can be difficult to scale for a couple reasons. There's either unsafe reagents, unsafe conditions, they suffer from low yields, low reproducibility, or they are isolating in trying to purify oils. And really at this time is when the process development chemist starts to strategize on how we can best scale up this process. Just a quick slide here for a health hazard classification. Health hazard classification is a numbering system that helps us quickly identify the potency of a drug substance. Here at Regis, we typically operate in the 2 to 3 category range. And I do want to point out here at the top, I've got Regis's banding system. Every company does have their own banding system. Below, you'll see the banding system for SafeBridge. One thing I do want to point out here is as we increase in potency, as we go from categories 1 to 2 to 3, the expense of the equipment needed to actually contain those materials in a safe manner also increases. So when we first begin doing lab work for process development, we begin with a batch that we refer to as a process familiarization. So what is the process familiarization batch? This is our first chance to run the client's procedure. And you can see with this first bullet point, it's really important that we run the process as provided. Typically, clients have a process that they've been working with for several years if they've gone through their own development. And it's really up to us to repeat that process as provided, and that helps us to provide a fair assessment of things that we need to change as we go to scale up. As you can see, one of the early themes as we go through this is that when we start, we're really making that eye towards late stage manufacturing and the things that we can do early on to help save time at those later stages. What's nice about the process familiarization is also at this time as we are starting to generate intermediates and product markers, both process chemistry and analytical development. These intermediate and product markers are important as it allows us to start working on our analytical methods and also helping to evaluate our data in the process that we have. During this time, we are also getting semi-accurate yield information, and we need that yield information to help us to begin planning production. It also helps us to figure out how much we need to start with for how much API that we're going to make. And we also use this time to really start gathering information. The more data that we have, the better that we can design our process to become efficient and scalable. One of the things I do want to mention here at the bottom is that we do begin to set process parameters. And what I mean by that is that typically we'll get procedures in that try to get us to heat something to 75 degrees. It's very difficult to hit something that accurate. And typically, we want to operate within ranges. So during this time, as we start to set our ranges, begin operating within them. At this early stage, this is a pretty loose set of process parameters that can be changed around as we get through development. During the process familiarization, a couple best case scenarios as we go through that batch. The reaction, the workup, and isolation perform as reported. They're nice and efficient, and they deliver the material as expected. Volumes are in line with Regis equipment constraints, and that is something to point out is that as we do develop, there are set equipment that we will have to run in as we scale up. So it is important that we design our process to be able to fit into those equipment. And when we get these client tech transfers, these volumes are already in line with our equipment constraints. And they also don't isolate oils. There's no column purifications or unsafe procedures. Worst case scenario is the chemistry fails. There's problematic workups and isolations. During the reactions, there are significant impurities that don't purge, which will have a critical quality impact on the final material. And there could also just be a lot of difficult to scale procedures, as mentioned above, with column purifications or isolations of oils. And typically, when we get these processes, they're usually somewhere in between these two. After we finish the process familiarization batch, we like to do what's called a control strategy. Control strategy is where the technical groups sit down and really either plan out or confirm that we have added enough control into the process. These technical groups can usually consist of process chemistry, analytical chemistry, and solid state chemistry. Process chemistry, we're concerned with reaction completion criteria. What does it mean when we say the reaction is complete? What happens if the reaction isn't complete? How can we fix that for remediation options? We also want to determine what in-process controls are needed, what purifications we think will be needed as we go forward through the development, what we tend to encounter for the workup and isolation process, and any intermediate or final acceptance criteria that we need to move the process forward. At this time, analytical chemistry is starting to think about the generation of the test methods that they'll be needing to develop. And they'll also be working very closely with process chemistry in developing our in-process control methods and looking at the limits of what we have with our analytical methods. Solid state chemistry is focused on the salt form selection. As I mentioned, we typically don't want to isolate any oils, and one of the ways we get around that is through salt selection. We want to look into drying studies, microscopy, recrystallization options, and polymorph detection. Underneath this control strategy umbrella, we've got in-process controls, process parameters, and specifications. And just a quick definition for in-process controls. A in-process control is essentially a control point where a yes and no event takes place. Really, based on the data, we either want to continue forward with the process, everything is working well, and we'd like to continue on, or we have a failure point, we need to do something else, we will resubmit it for that test will pass, and then we can carry it forward. And that is one of the ways that we ensure that we will get the quality needed out of this process that we intend. Some common questions that get asked and answered during these control strategy meetings. When is the reaction complete? What does the reaction complete means? It's very rare that we have starter material go to product in 100% efficiency. So we need to know what exactly it means when the reaction is done. What happens if the reaction stalls? How do we know that the solids are dry, or does this solid filter well? As we begin, so after process familiarization, we've gone through our control strategy. We have a plan in place of how we want to approach development. The initial development approach starts with vial scale reactions. As you can see, we have a picture of a typical stir plate with a couple experiments going there. These are typically agitated with magnetic agitation and stir bar, and this is important. As we scale up into production, we don't have magnetic stirring, we have overhead stirring. So with these vial scales, really what we're looking for is reaction and workup screening. We run a lot of different experiments. These are quick, and we're just looking for, does it work? Is this looking good? Things like that. We also look into the reaction profile, and we start to get a case, even at this early stage, of what kind of impurity information are we looking at? Is there a major impurity that we need to purge later? Is the reaction fairly clean? We also use this scale to look into solubility information. However, these vial scale reactions are such a small amount of material that these aren't very useful for getting yield information. You don't get any consistency between your reactions, and it's hard to go with robustness for these vial scales. So after you do your vial scale experiments, we do our first scale up. These are going to be in round bottom flasks. Typically, the smallest quantity we start with is 5 grams, all the way up to 50 grams, possibly a little higher. But the key here is that we switch into overhead stirring. We were using those vial scale experiments to really kind of figure out what process we think will work pretty well. We then go to this overhead stirring, and now we're starting to look in reproducible reaction results. We also want to define all parameters, and we want to stay within those parameters. This helps us to determine if we've got consistency with the process as we've developed. We also want to look into the efficiency of workups. Typically, the reactions are a small part of the actual chemistry that we need to do to go from start to finish, and a lot of time will be spent on making sure that we have an efficient workup, and more on that to come later. Also at this scale is where we're really starting to dial in our yield information and to confirm that we do have consistency with our process. Here I just have a picture of our EasyMax setup. So this EasyMax is a programmable piece of equipment that really allows us to have tight temperature control. We can even program different temperature plans throughout a reaction or workup process. Off to the right there, you'll see that we have an addition pump that allows us to look into slow addition of reagents or solvent additions. And these are really nice to help us work on one of the more difficult things that we deal with in process chemistry, which is designing a crystallization for good purification of our material. We can also use this equipment for process safety with calorimetry and get a lot of thermal hazard data on our reactions. The second scale up. So here you can see we've increased in scale. This is a 5 liter jacketed reactor. And here what we're looking at is that now the mixing and the process is getting really close to what we can expect on the production scale. Here we are starting to draw out reaction times, addition times, and hold times to more closely mimic production runs. And I want to point out that certain events just take longer as you go up in scale, and this is when we really start to probe those timings. Here we also want to confirm our mass balance. At this point, there is a significant investment in material, and we want to be sure we know exactly where that material is going, where we're losing any of the material, and that kind of thing. And as I mentioned before, this workup here at this stage will really closely resemble the production for the extractions, the washes, filtration, and any crystallization conditions that we have. I want to spend a few minutes going through the workup and product isolation. As I had alluded to earlier, a typical process out in our production facility will generally take 3 to 4 days, but the reaction is only a couple hours of that part, with the rest of that time spent on to get the product out of the reaction solution and to isolate the material. And the goal of the workup and product isolation is really to get the material out of the solution in an efficient way. And by efficient, what I mean is that by both time and effort, a process that is a little tricky in the lab can easily become a several-day painstaking effort. During this time for the workup and product isolation, we're really interested in, can we remove impurities? If we're originally we're generating oils, can we make it into a salt so that we now isolate solids instead? If we need a purification step in there, that we're now looking into crystallizations rather than column purification. And we also want to make sure that we're getting out the correct solid state form. As Ron alluded to earlier, it's really important. And as we go through development, we are changing temperatures, we are changing solvents, hold times. We're changing a lot of the process, and we really want to be sure that we are generating the form that we need for these materials. At this time, we are also developing any isolation and drying conditions we have. And I just want to point out that the theme of this talk is the integration of solid state chemistry with process development. And really, for all of those goals, as we go into the workup and product isolation, involve heavy collaboration with solid state chemistry. I just want to go through a couple of commonly encountered problems during the workup. Probably the first one is emulsions for everybody. And just a quick definition of what an emulsion is, a mixture of two or more liquids that are normally immiscible. And really that doesn't do it justice. If you look at the picture on the right here, an example of an emulsion, you've got a top liquid layer, a bottom liquid layer, and then in between, we have a third something layer. In that layer can be product. Typically, there's solids, there's oils, there's particulates, and it's really hard to deal with. This event in the lab is only probably about half an hour, 45 minutes for us to find a way around that. However, out in production, this could lead to several days as we try to figure out how to get material out of that cleanly. Another thing that we deal with is multiple extractions. A typical process will just have 3 extractions. However, when we scale up, 1 extraction is about 4 hours in the plant. So we really want to minimize extractions and only limit to the ones that we need to actually move material into the phases that we need. We typically deal with at this time too, as I've alluded a couple times throughout this talk, is that the reaction solution is being concentrated dryness to give a viscous oil. How do we move that viscous oil from one reactor to the other, or how do we even get it out of the reactor? We very well can't pick up these 500-gallon reactors to pour the material out like we can in the lab. We also tend to deal with a lot with slow filtrations. Slow filtrations in the lab, a couple hour event for us. Again, this really translates to a couple days to even a week of filtering material out in production. I just want to have a quick example here of this is a workup and product isolation. So this here is an extraction. Here you can see there's 3 different phases. On the previous slide, we had an emulsion. Here is actually a clean middle layer. So we have a top organic layer, a middle unreacted starting material, and a bottom aqueous. What we found in the lab is actually really nice is that you can remove that middle layer and it actually acts as a purification of the material. You just have to closely cut those layers. Unfortunately, when you go to scale this up in production, you can't see inside these vessels. And it's much harder to see where those phase separations are. And you can see our only site is that small area at the very bottom of the reactor. So this is an example of something that looks good in the lab. We scale up, we see those 3 phases, and then at that point, we go back into development to try to find a different way around this that will be an easier and more efficient way through. I want to take a brief moment to talk about what a systematic approach to process chemistry means for us. And really what we're looking for is we want to attain similar results across a few different batches. We want that consistency. I'd mentioned that when we first start, we have vial scale reactions. There's multiple reactions, multiple workups. Material is all over the place. So really, once we figure out a good process, we do that first reaction. So we take that first lot of starting material, which gives a product, and that's our initial result. We've got purity and we've got yield at that point. The next step is take that exact same lot of material, usually run this process at the same scale and make sure that we get the same yield and purity out at the end, and that gives us our reproducibility. We then like to take a different lot of that starting material, go through that process again, and make sure we do get the same lot, and that's that 1-factor variability. And this lets us know that we can use different lots through the same process and get the same result out. And the third and probably most difficult one is to use a third lot of material, scale it up, and then hopefully that gives us our same result. If it doesn't, then we tend to go back into development and try to figure out where the issue came from. I want to go through hold points and stress tests briefly. So what are hold points and stress tests? So a hold point One of the overlooked aspects of scaling a chemical process from the lab to the plant is that everything can just take longer. How sure are we that the product is stable at various points? Is it okay for the reaction to go another 12 hours? What if you hold the product solution over the weekend? What if there's a holiday? These are real questions that have been asked throughout my career. And early on, it usually starts with, I'm not sure, let me go back and run that test. We try to answer those questions early in development if we can, in preparation for those questions. For stress tests, a stress test is when we purposely force the process outside of the normal operating ranges. This is something that gets really heavily looked at and investigated during late stage development. But even in early stage development, there's a few parts of the process where we can just be nervous, and really stress tests help the process chemists sleep at night. What if the in-process reaction fails and we need to heat the reaction longer than intended? If we've never heated it for that long in the lab, that's a point of concern. So during development, we do these stress tests. And as I mentioned, during late stage development, you really look into that to make sure that you have robustness for your process. After we get through development, our next batch that we have is the lab confirmation batch. So the lab confirmation batch is there for a reason. Typically during development, we have generated multiple lots of materials, run through multiple reactions. And the lab confirmation batch is really that batch that we go start to finish through the process. This is the first time that we do that, and we generally only make minor tweaks to the process, and most of the tweaks that we do make are mostly mechanical in nature and not chemical. This is where we're starting to change how we do things, not what was going into the vessel. And this is basically the first practice run for the process. This lab confirmation batch is a joint effort. Solid state chemistry is involved for solid isolations, making sure that we're getting the correct form. Drying conditions are adequate and more. The tech ops group, they're essentially the liaison between the lab and the production facility. And as I mentioned earlier, you can't very well pick up and tip a 500-gallon reactor. So they'll start to look at the process and add some changes that we need so better mimic of what we can actually do out in the manufacturing floor. At this time, analytical development is also it's also a part of this. Essentially, as we are doing the development and we're doing our first practice run, this is the first practice run for us to look at the analytical methods that we have. Are the in-process controls that we have developed adequate? Do we need to make some changes? We want to go through that before we transfer them to our quality control group. During this time, we also talk about process safety, and we are doing thermal hazard assessments of our process. After the lab confirmation comes the demo batch. After lab confirmation is complete, the next batch that we run is the demo batch. The demo batch is where we start to test the scalability of our chemistry. It usually is a significant increase from the lab confirmation batch and somewhere in the 1/10 of the scale of our next planned production batch. As I'd mentioned, the purpose of the demo batch is to test the scalability of the process. And this is the closest batch in terms of equipment, along with stir and hold times that we'll use in production. These batches are typically non-GMP, and the material generated will be used as reference standards for an upcoming GMP batch. And really, we'll take the results of the lab confirmation and the demo batch, compare them, and we'll use them to set our specifications going forward into the production batch. So what is our role during the production batch? For process chemists, we are the subject matter experts, and typically we'll be present from the batch from start to finish. We'll be on the floor and we'll be present during the entire process. It may no longer be our hands running the actual process, but we are very closely involved at this stage. Sometimes unexpected issues arise, which require a solution, and this will happen typically during pretty early phase work. And at that point, the process chemist will be taking samples back into the lab, developing a path forward, and then we'll be implementing it in production. Just a very brief slide here on what is late stage development. So late stage development can be an entire talk on its own. And here I just wanted to point out a few different things. As we go through the development and the early phases, we start to go into optimization. And during late stage development is where we switch over from optimization to understanding. Typical activities that occur during this time is we start to look at a proven acceptable ranges. I have a quick graph here on the right to kind of show what that means. In the gray box there is where we intend to operate. However, we want to know that if we deviate just outside of that range, how safe we are that the process can continue and deliver the quality of goods that we intend to make. During this time, as I mentioned at the very start of this talk, we are looking at impurity, fate and purge studies. And this is, where are we generating our impurities? What are the key impurities? Where are they going? And what levels can we tolerate? During this time, we also determine our critical process parameters, which has a direct impact on the quality of the material, or the determination of key process parameters, which has a direct result on yield of our process. At this time, I'd like to turn it over to my colleague, Chris Castle. He'll be going through several case studies to kind of go through the topics that we have covered here so far.
Thank you for that lead in, Eric, and good afternoon, everyone. So as we round off this talk with a few case studies, what I wanted to bring to the forefront is to illuminate a common theme that is going through all of these case studies. In fact, actually two of them. I want everyone to be mindful that these case studies were representative of collaborative efforts between process chemistry, solid state, and analytical chemistry to identify common problems and some not so common problems, and also to work diligently to ultimately improve our processes. And another aspect that I wanted to emphasize and that these case studies will illuminate is that the reaction and the success of a chemical reaction is only a very small part of the larger chemical process, which I hope that we'll underscore here. So for our first case study, ultimately, our theme here is that we are engaging solid state chemistry to dynamically read quickly, solve a common problem that's associated with scale up of chemical processes. So for this particular example, we had an intermediate material that we were isolating on the route to an API. Regis had successfully manufactured one tox batch, and then we were doing process development that had initiated prior to our scale up to a first GMP campaign for phase 1 clinical studies. During our development, we had done, as is our process, we had done several experimental runs, which were completed immediately prior to lab confirmation. And our findings were that the yields were consistent and the purity was about level when we compared one lot or one experimental run to another. So we had quite a testament to reproducibility. So we felt fairly confident going forward there to scale up to the lab confirmation batch. And as we ran the lab confirmation batch, we discovered that the purity remained consistent and the yield increased. So we could sort of maybe pat ourselves on the back. Life's great. You would think so, but not so fast. So we had to check one of our assumptions because as we found out, as we carried this material forward into the next step, the reaction ultimately failed with large excesses of starting material and a key reagent left over. And it sort of left us scratching our heads as to why that would be. So to answer that question, we go back to the lab. And then we evaluate, we do some experiments and we figure out what is the data actually telling us. First, from our observations, the purity remained consistent from one lot to another by HPLC analysis. We saw no significant variance from one lot to another. And then a quantitative analysis using two separate analytical methods, in this case, NMR and HPLC, showed that the weight percent assay or the overall potency was indeed low compared to other lots. So we had good confidence in the veracity of this result using two orthogonal methods to analyze. And then our more conventional qualitative assessments by proton and carbon-13 NMR still appeared normal, no unexplained peaks. So the question becomes, what then? Is there something invisible to HPLC, NMR that we're not seeing, something inorganic perhaps? And there are some tools that you could typically use to arrive at an answer. One analytical tool that might come to mind might be residue on ignition, ROI, which is, unfortunately, it can be a little bit time consuming to obtain a result, and it won't actually tell you what you have from a definitive standpoint. It'll just confirm that indeed you have something inorganic in there that is lowering the overall potency of your lots. So what we did is we engaged our analytical and solid state expertise, and we decided to try X-ray powder diffraction. So with the help of our colleagues in solid state, we submitted a sample, and then within 1 hour, we had our answer. And the spectrum that you're seeing at the right should be fairly common, or should be fairly recognizable to people who have seen salt before. So what we were seeing was sodium chloride that was ultimately trapped in our intermediate. And one point I want to emphasize here is that sodium chloride has a very distinct spectrum by X-ray powder diffraction. And if you have this impurity mixed in with your solid materials, you can tease out all of these peaks, and you can compare it to a reference, and you can indeed say you've got sodium chloride. But where was the source of the sodium chloride? It made sense from our perspective because we had used sodium chloride in saturated brine washes during our workups to aid in phase separations. And when we discovered that this was an issue on scale up, we immediately just wanted to see if we could maybe cut down the sodium chloride loading and make a more dilute brine solution, and then try the washes at higher temperatures, which was successful ultimately. And we would see successful phase separations. And then ultimately using the strategy and going forward, we saw no further problems with the overall potency. And ultimately the chemistry of the next step was successful. So this was a particular example of us engaging our subject matter experts in solid state in order to quickly and efficiently solve a problem that we were dealing with on scale up. Now, as I take you through the next couple of case studies, again, I just want to emphasize that the reaction and the success of the reaction, the completion of the reaction, is only a very small aspect of process chemistry. In fact, you might even just call it the tip of the iceberg. Where you really start having to run into considerations as far as cost and time, really focused on workups, isolations, filtrations, typical unit operations that you would see performed on large-scale manufacturing. So for this next case study, for this example, we had a procedure that was provided through a technical transfer from a client, which we investigated in the lab, doing our due diligence during familiarization, and then ultimately extrapolating that into some mild scale-up work and early development. For this, the desired product is isolated after several extractions, and those extracted layers, organic layers, are concentrated in one process stream to near dryness, and then the dried material is slurried in water. And then the isolated product that we obtain is an amorphous powder. It sounds all well and good to actually describe it in words, but I thought, as is my typical habit, I sort of like to map these things out. So I wanted to actually illustrate that on the next slide. So this diagram is an illustration of that process as it was provided to Regis. So I'm going to pause briefly while I give you a couple of seconds to take a look at this. Now, what I want to emphasize here is that you've got your product in the top left, and ultimately from the time you have your reaction mixture in the top left to the point where you go to your isolated product in the bottom right of the screen, there are a lot of unit operations, in particular, a lot of extractions, a lot of phase separations. There are multiple waste streams. There's ultimately concentration to near dryness under reduced pressure, and you find yourself wondering how you could possibly scale this. As Eric had alluded to earlier in the talk, when you are in a situation where you are doing extractions, they can take several hours for each extraction for all of these operations. And realistically, when performing this particular procedure in the lab, this might be about 2 hours worth of actual effort from the time that you are in your reaction mixture to the time you have isolated product. But this could translate into realistically a week, if not more, in the plant, to say nothing of how much more time that you would have to factor in when you consider varying levels of scale up. So ultimately, the problems that we identified early on with this process is that you have multiple solvent extractions, and as you scale up, this would equate to more plant time and costs, especially when it relates to waste disposal. We have to consider the concentration to dryness and neat oils, which is not in itself amenable to scale up. And the yields, in all likelihood, will not be consistent as you scale up, because as your scale goes up, your material input goes up, your extraction volumes go up, and you also have multiple streams where you could be potentially losing products. So you don't really demonstrate. There's not a lot of potential for control in a process like this. Needless to say, when we saw this in the lab, we just thought there's no realistic way that we could scale this up. So we started looking and casting about for some other solutions. So our findings in this particular case study is that in the legacy process that we received, isopropyl acetate is just used as an extracting solvent from the reaction matrix, which is ultimately an NMP water. It's only concentrated to near dryness, and then we isolate a product that is typically amorphous or poorly crystalline, which is not desirable on scale. And I could say a lot more about the crystallization of organic compounds from an industrial perspective. It could fill a pretty decent webinar in and of itself. I wish I could go into it a little bit more, but suffice it to say, amorphous solids don't offer predictable filtrations or impurity purges, and ultimately it's a poor control on scale. Ultimately, you'd want to see if you could actually isolate a crystalline material. In this particular case, we were able to take some of the material that we had isolated from this older process, and we found that we could actually recrystallize it relatively easily in a mixture of water and NMP, which is more closely related to the ultimate reaction mixture. So we go from amorphous to crystalline in this matrix with one consistent solid form. So microscopy data on the bottom, the picture on the left is actually showing the amorphous material that we had initially isolated with varying particle sizes. It clumps fairly easily. It's relatively difficult to filter, and it might be between the varying particle sizes, and it doesn't afford you a great deal of control or confidence going forward. But as you go from left to right, you do the recrystallization, you find that you have these beautiful plates that filter very nicely on lab scale. And we, with some mild tweakings to the reaction process and the isolation process, we reworked the procedure to something a bit more amenable to scale. So the process here is greatly simplified, where we have our product in a slightly tweaked mixture of NMP water. Ultimately, we add a bit of water to it to dilute, and ultimately we isolate a slurry. Or sorry, this results in a slurry. We installed in-process control to demonstrate some degree of control over of our losses and the supernatants and the mother liquors. And the beautiful part about this process is that, yes, it is greatly simplified, and there's also only realistically one major source of loss for the product, and that ultimately translates to whatever is lost in the crystallization in the mother liquor. So our improvements from the old process to the new is that we've now reduced the waste. We've lowered waste disposal costs. We have an extractive workup, which would have been quite difficult to perform on scale, which is ultimately reduced, or I should say replaced, with a dilution and filtration, translating to fewer operations to perform on scale, overall greater efficiency. And then we end up with a crystalline product that we can purify further, and we can also recrystallize if needed. This actually translates fairly well into the plant. As of now, we've done several campaigns where we've isolated typically 73 to 75% yield with greater than 99.5% purity for this particular case. But this was a nice example also of process chemistry engaging with our subject matter experts in solid state to quickly identify crystalline forms and to maybe look at workable solutions that could translate well into the plant. For this final case study, we're also focusing on the workup, but in a sense, we have an enabling route that we initially developed at Regis to access a final API for phase 1, phase 2 clinical trials. The emphasis for the first round of development, as it typically is on time and expediency. You're just trying to get the material out there and get it isolated. And you may not necessarily have as much of a mind to optimization at this point. It's a matter of, can we have something that's scalable and safe? But we were also successful in this initial process on having relatively, quote unquote, simple isolations that avoided column chromatography. And the step that we're concerning ourselves with is step 3 in a 5-step sequence. So for the phase 1 process, the legacy process, the reaction was completed in THF, which is a water miscible solvent. We ultimately filter to remove some minor byproducts, and then we concentrate the filtrate to an oil, which is then, we then, after concentration, there's a sequential precipitation of salts and byproducts through the addition of some non-polar solvent, and then there's a second filtration, and then finally the filtrate is concentrated again. We reduce the volume and then add yet another solvent, an anti-solvent in this case, and heptane, and then we cool. The product precipitates, we filter it, we collect it, and we have our isolated product. The problem with this process as it stands is that There are several salts and byproducts that precipitate and crystallize out with product, and we see some relatively unpredictable product loss. There's lots of concerns over mass balance and the reliability of this, and we would have more concerns about continuing to scale a process such as this. As far as the unit operations are concerned, we have 3 filtrations, 2 solvent swaps, a concentration to an oil. Ultimately, this boils down to more concerns about time, operations, and the quality of our material. And the yields are variable, 30 to 45%. But as our projects and as our clients advance their drug candidates in the clinic, and it continues to do well, so too should our processes and our understanding of the chemistry improve and mature. So with that, we actually went back into the lab and we did some critical reevaluations of the process and what we were looking at. And despite some of the challenges in phase 1, there were some bright spots to be happy with. First off, again, leveraging our expertise of solid state chemistry in-house, in particular X-ray powder diffraction analysis, showed that several GMP lots isolated under legacy conditions showed the same crystalline solid form. So we could look forward to some consistency there. And consequently, in the lab, when we were screening various solvents and anti-solvent combinations, we were also seeing the same solid form. We knew that the byproducts of the salts would be a problem for removal, but we also knew that they were water soluble. If possible, we would actually seek to remove them with water washes and try to minimize losses of the product along with it. So our natural inclination went to switch from a water miscible solvent for the reaction to be completed in to a water immiscible solvent. In this case, we made a switch from THF to methyl THF for the reaction. Once we established that the reaction could proceed fairly well in methyl THF and the crystallization could be done in methyl THF and then heptane, this gave us the tools that we needed to modify the process. And after some due diligence in the lab to scale up, ultimately, our process now evolved to one where we've now completed the reaction in methyl THF. The organic stream is washed with water, and then the washed organic stream is still concentrated to a nominal volume. And then we charge N-heptane as an anti-solvent. The slurry is cooled for about 1 to 2 hours, and then ultimately we filter and we collect our product. It sounds simple enough on the face of it, just to simply add in a water wash. But when you boil down what the implications were for this particular project, it's actually quite striking. So as of now, first off, the operations for water washes are successful in removing the salts and the unwanted byproducts, and ultimately we have less than 1% product loss in the washes, which was a quantifiable and consistent result. And then ultimately our main source of product loss was through the mother liquor. So this was again quantifiable and consistent, affording us greater control. To this end, we have successfully executed 4 GMP campaigns using this process. We've improved our yields from 30 to 45% to about 74 to 75%. Unit operations, for one example, we've reduced 3 filtration operations to 1. And we've reduced solvent waste, and we removed 1 unnecessary solvent from the process. And ultimately, this translates to process improvements that, when you look at the numbers, they afford about twice as much product for the same input of raw materials. And it was actually this extra throughput that really just generated, in this particular case, an added value of about $1.5 million to our client. And this was just for what Regis has manufactured up to this point, to say nothing of future manufacturing campaigns and the extra value that we would provide. So to that end, we were ultimately satisfied with the collaboration and the working together of solid state and analytical and process chemistry to generate a practicable and workable solution that translated to a real value for Regis and for our clients. In conclusion, I really just want to bring this back around to say that we've had a more all-encompassing talk about process chemistry at Regis, and we just want to emphasize that overall process development is the development of a safe and consistent and robust chemical process. And even though we of course care about the reaction that we're we're running. The reaction chemistry and the completion of a reaction is only one small part of a chemical process. And process development ultimately has to keep the end game in mind. So how our process will scale into GMP manufacturing, in particular Regis equipment. And then process development ultimately is not a siloed event. It's a dynamic coordination and collaboration with an interdisciplinary team of subject matter experts. In this case, our key players were process chemistry, solid state, analytical chemistry. And we also just want to emphasize that engaging these teams and working together early can translate to a higher return on investment for our clients and ultimately a greater understanding and mastery of our chemical processes, which we hope to carry forward. And as an old saying that we're adapting, we're shamelessly adapting for this presentation to say an ounce in the lab saves a pound in the plant. So with that, I really just want to thank everyone for their time. And I think the team would like to open up to any questions you may have.
So, all right, this is Dan again. Again, thank you. I see we did have a few questions that were typed up in the chat. We have just a few minutes here, so maybe we can get through one or two, and we certainly will take the rest of the questions, and maybe we can respond back to those offline and set up some time to talk through those. I think one of the first questions we saw here, and maybe I can pass this one on to maybe you or Eric or Chris can take. Do you set a limit on impurities when you initially start the process development? And maybe Eric, you can take that.
Yeah, sort of. So when we first start process development, we typically have a ballpark of what we're looking for. And honestly, generally that number is about 95%. But when we first start development, you're kind of looking to see just what we are getting out with the process. And really, as you go through the different phases of development, that number typically goes up. Even later stage, we have intermediates where we're looking for 90% purity, but that's because we know downstream in the process that we do get that impurity increase to what we need to deliver ultimately at the final end of the API. So you generally start with limits, you move them around as you develop the process.
All right, we have another question. What is the best way to isolate compounds which are low in melting or oil in nature?
Yeah, so to answer that, without knowing a lot of information about the molecule itself, two ways that come to mind is one is temperature control for your final isolation step. Another is whether or not you can generate it as a salt or a co-crystal and isolate it that way. And again, depending on what your end product should look like, really will dictate how you isolate that material. But that at least without knowing a lot more, those are two typical approaches that you can use for low melting compounds that you're trying to isolate.
All right, it's actually starting to look like we're actually running up against the hour here. I do see a few more questions. Perhaps if there's a little time, folks can stay on, but I do understand people are probably going to be dropping off shortly, and we have some things to do. So maybe we could take one more question. There was a question regarding how would these process development strategies apply, and could they apply at another CDMO besides Regis? Maybe Eric, if you could take that one.
Yeah, I can take that one. Absolutely. And honestly, if you talk to any process chemist, we all do some sort of this approach to develop our process to have that scalability, safe and robust process. And really, this is just a strategy that we've worked out for the success of Regis. But as I'd mentioned, really most of us do this. And at the end of the day, the last thing you want to do is gamble as you scale up. So really running those batches multiple times, having meetings with your technical group, talking through the process. These are all things we all do to make sure that we have that robust, consistent, and safe process.
I think we hit, I just want to make sure we put up our contact information here before everybody moves forward. So if, there it is. And so like Lauren and I mentioned at the beginning of the presentation, if you have any further questions, you want to reach out, talk about the content of this slide or any of the other talks that we've had for that matter up to this point. Here's our contact information. Please feel free to reach out. Lauren, if there's anything else you want to add before we move on.
No, other than, feel free to email us with any of your questions, and we will be wrapping up this webinar series with a Q&A session where anyone can come with any questions, regulatory, CDMO, CMC related, any questions that you might have in the development of your program. That will be posted in the near future, probably in Q1 of next year. So keep your eyes out for that. It has been a pleasure to put this together with you guys, and I hope that many of our viewers have found a lot of education behind it.
Good.
Right. Yeah, thanks, Lauren. And there has been a recording, so we're going to try to make this available to everybody if you want to catch up and go back and pick something out. Again, thank you everybody for attending. We're 2 minutes over. I want to be respectful of all your time, and hope to see you all again soon.